Good morning or afternoon, everyone. I want to welcome you to this webinar. I'm Christy Sullivan. I'm the Secretary of the American Society for Cellular and Computational Toxicology. We are presenting this webinar jointly with the European Society for Toxicology in vitro because we share a lot of commonality with our European counterparts. Both of our organizations hold regular meetings and pursue other activities to promote computational and in vitro toxicology. In particular, we offer training and education opportunities and aim to support early career scientists and interaction with scientists from different fields, regulators, industry, academia. I want to give a couple of brief announcements first before we get started. The first is that there are 2 meetings in June, depending on which side of the Atlantic you'd like to be on. We, first of all, the ESTIV International Congress, which happens every 2 years, is going to be held in Barcelona this year. And you can visit the link to find out more about the conference and register. We are also involved in promoting and helping to host the 19th International Workshop on QSAR, Environmental and Health Sciences. That's going to be in Durham, North Carolina, also June 8th through 11th. And we have a really nice, robust program that's just about to be published. And there's a late-breaking abstract opportunity for a couple poster slots that are still available up until April 8th. So please consider participating in that conference. As far as we are intending, those conferences are still going ahead, but of course we'll keep monitoring all of the travel situations. And finally, I'm really happy to announce that we have dates for the 9th annual ASCCT meeting. That meeting is going to be in October, also in Durham, North Carolina. And the theme is focused on the EPA goal to reduce reliance on mammalian testing and what are the needs to make that happen. We have 2 confirmed plenary speakers just now, Dr. Richard Kegwin, Office Director of the Office of Pesticide Programs at EPA, and also Tara Barton-McLaren at Health Canada. And watch out on our website, abstract submission will open up really soon for that. And finally, we don't have any upcoming webinars for the first time in a long time. We have these webinars monthly, and we have an open spot for next month, April. So, maybe you have already prepared your Society of Toxicology presentation and you want to be able to share it with your colleagues. I would encourage you to get in touch if you either want to give a webinar to the ASCCT and ESTIV community, or you would like to nominate, maybe a topic that is of interest to you. So please get in touch with me. My email is ksullivan, that was on the previous slide there, or you've, I'm sure, received emails from me before. So finally, just to do a little bit of housekeeping, this webinar is recorded. ASCCT and ESTIV members will have access to the recording shortly after the event, and everyone will also receive a PDF copy of one of our presenters' slides today. Your lines are all muted, and there will be an opportunity to ask questions after each presentation. And so please, as the presenters are speaking, feel free to enter your questions so that once they're finished, I'll be able to read them out and we can have the Q&A session. We have a really special webinar today highlighting ASCCT award winners from 2019. During each annual meeting, 2 presenters are selected to win 2 awards. One of them is the Ed Carney Predictive Toxicology Award, And that was awarded to Ignacio Tripodi, our second presenter. And it was established in 2015 to honor former ASCCT member and toxicologist Dr. Ed Carney. And our first presenter, V. Tran, has won the Tox 21 Student Award, which was endowed by ASCCT member Ray Tice in 2016. So without further ado, I'm going to introduce our first presenter. While I'm talking, I'm going to try to give her control. So Ms. Tran is a PhD student at Johns Hopkins University Bloomberg School of Public Health. She obtained a bachelor's degree in biology at Georgia State University in Atlanta in 2017. And she is working at the Center for Alternatives to Animal Testing under advisor Dr. Thomas Hartung. And her current research focuses on using bioinformatics and publicly available data to understand cancer. Her thesis project is about predicting novel transcriptional factors and modeling of iron metabolism in gliomas. So please go ahead. I see your slides. So you're all set to go.
Thank you, Christy, for the lovely introduction. It's my great pleasure to be here. Today, I would like to talk about using network analysis to predict, to compare the difference and similarity between the breast cancer cell line and human breast cancer tissue. So to start with, I would like to ask a question. So if you could help prevent the misuse of tens of billions of dollars in research, would you do it? Because currently there is a high prevalence of irreproducibility in research, which is estimated to be 50%. And to back up that statement, here is a number of reports on the prevalence of irreproducibility in research, ranging from both industry and academia, and the estimation is between 51% and 89%. And this inefficiency results from a lot of loss of money. The estimate is that there is $28 billion a year that in the US that is spent on non-reproducible preclinical research. And this also can have a lot of financial implications on downstream clinical trials. In particular, it is safe to say that we are currently in a reproducibility crisis in cell culture. An estimation shows that there is a 36% of misidentified and contaminated cell line, and another study from Science estimated that 20% of cell line that is currently used in research is cross-contaminated. A quick look at the NIH site shows that there are about 9,000 projects that is actually using cell lines. So clearly cell culture plays a very important role in biological research. And the sad thing is that although methods to authenticate the cell line and quality control method are widely available and at a relatively low cost, not many researchers are using this method. A survey found that there's only 25% of researchers that are actually using authentication techniques. So perhaps many of you here are familiar with the HeLa cell story. So HeLa cell is a cancer cell line that is taken from a woman with cervical cancer from here in Baltimore, from Henrietta Lake. And this was the oldest and most widely used cell line in research, although it has provided tremendous value in research. There is a lot of genomic instability. A study published in 2015 showing that a different batch of HeLa cell express different percentage of gain and loss in genomics compared to human genome. And imagine that if scientists use HeLa cell, like it would be very difficult to get reproducible results. So my project today focused in particular on the MCF7 cancer cell line. This is a human breast adenocarcinoma cell line that was developed in the 1970s from the Michigan Cancer Foundation. And this is among the most commonly used cell line for in vitro cancer research. A search on PubMed will show that this cell line is featured in over 30,000 publications, and it also involves multiple subcell lines. So previously, we have done a study on MCF7 cell line in terms of its variability from our lab. And here is an imaging picture showing the MCF7 cells that come from the exact stem cell batch from the stem cell bank that we did in parallel between our group at Johns Hopkins University and Brown University. And you can see that the morphologies of the MCF7 cells from the two cell batch are quite different. The Brown University cells on the left show more round and aggregated morphology, whereas our cells from Johns Hopkins University has a more flat and cobblestone morphology. We also measure MCF7 cells response to estrogen stimulation. And the data is showing that in terms of estrogen response and estrogen agonist response, the group in the MCF7 cell line in Brown University show a much stronger response. And in the panel at the bottom, we also measure the the gene expression of the estrogen receptor, progesterone receptor, and it's showing that the Brown University MCF7 cell also has a much stronger response. So the cells come from the same cell batch, and with this data, we are showing that even though with standard quality control methods, there can still be variability in MCF7 cell. Another issue is the clinical relevance of cancer cell lines in research. So not all cancer cell lines have equal value as a tumor model. And to address this problem, the NIH has developed cancer cell line panels for drug testing. And although these panels have generated wealth of information, but it's also little confusion in terms of the origin of the cell. And it also increases the noise in the data. So based on this gap in the literature, that is unknown whether the transcriptomic landscape of MCF7 cell is similar to its tissue of origin, and there have only been a few studies that directly address the relevance of MCF7 cells as a model for cancer drug testing. My project had two aims. So first, I would like to identify the key similarities and difference in the gene expression profile using network analysis of MCF7 cell as compared to its tissue of origin, human breast cancer tissues. And the second aim is to determine the value of MCF7 cell as a cancer drug testing model. So for the first aim, I'm using two main data sets. So for the MCF7 data set, I'm using a pool sample data set from ASCCT database with over 1,000 samples. And the human breast cancer data set is from Genome Cancer Atlas with also over 1,000 samples. So these are both relatively large data sets. In terms of analysis workflow, so we started with doing data preprocessing by removing the missing value and perform normalization, removing outlier as well, checking for batch effect in these data set. So the final data set that we have is 915 samples for both MCF7 and human breast cancer data. And to avoid low expressed genes that will not contribute to biologically meaningful implication, we filter for the top 1,000 most variant genes in terms of its expression. And then we perform weighted co-expression network analysis, which is a clustering method that I would go further into detail in the next few slides. And then after that, we perform consensus analysis to really compare the similarities and difference in the network between MCF7 and breast cancer cell line. So weighted gene co-expression network analysis is a graph theory-based approach, and basically it works to cluster genes into modules. And this is an unsupervised learning approach, and we have been using this method for a number of studies that have been previously published. So for weighted gene co-expression network analysis, or WGCNA, to work, you first start with a gene expression matrix with the column correspond to expression value and the rows correspond to a gene. And from there, you can construct a similarity matrix that is using correlation. And in this case, I'm using BiCore, which is a method that is more resistant to outlier compared to other correlation methods such as Pearson. And then from there, you can construct an adjacency matrix to determine the modules. So overall, the advantage of WGCNA is that this is a good by association approach to cluster genes with similar expression into similar module. And this is using a soft threshold approach, so it provides a more robust result compared to unweighted network method. And finally, after the modules is created, you can also visualize network and relating the network information to external information. So this slide is showing the number of overlap genes between the MCF7 dataset and the human breast cancer dataset, and we just constructed a very simple co-expression network. And we're using different threshold level, and basically this is showing that at different thresholds, there are minimal overlap between MCF7 and human breast cancer tissues. So to build up on that, as mentioned before, we did WGCNA analysis, and then we perform a consensus analysis. So about consensus analysis, it is to show the correspondence between the module from the MCF7 network or the modules from the breast cancer tissue network to the consensus. So on the left, for the column is the module. So each color indicates a module from the consensus analysis between the MCF7 dataset and the human breast cancer dataset. And the row of the figure on the left shows the modules that we got from the MCF7 specific network. And the color between each box in each part indicates the number of the overlap between the consensus network and the MCF7 specific network. And as you can see, there's not much genes that are overlap between the two. And there could be many reasons for this. This could be attributed to the variation within the MCF7 dataset itself. The ASCCT database collect sample from publicly available database for MCF7. So it could potentially include knockout cell life MCF7, and which can contribute to variability in this data set. However, for the figure on the right, which is the result between the human breast cancer tissue network and the consensus network, there is a lot stronger overlap. And I think this is because for the breast cancer data set, we extracted from the Cancer Genome Atlas, so it has higher quality data. So to back up that, we also are showing the results of our clustering for the MSEP7 dataset and the breast cancer dataset separately. And if you look at the bottom of each figure, the color indicates the module, and the gray color indicate that genes that cannot be assigned into any modules. And for MSEP7, this is clearly the majority of these genes. So again, this is supporting that the issues is staying with the variability within the data collection process itself for MSEP7. So for the next step of my project, I would like to determine the value of MSEP7 cells as a cancer drug testing model. So for this, we identify the hub genes within the network of MSEP7 and breast cancer tissue. And then we determine the ligand druggability score of the hub gene in each network. And this is using cancer database, which is currently the largest drug knowledge base for cancer drug screening in the world. So for the figure on the right, the top figure is showing the hub genes in the MCF7 network, and the y-axis shows the score. So the ligand druggability score is determined by using small molecular that have been tested against a protein or its homologue, and they calculate the likelihood that a protein is a target for drugs versus non-drug molecule. So for both MCF7 and BRCA datasets that are hub genes with a relatively high ligand druggability score, there is no overlap in the number of genes with the high druggability score. And this is interesting because if you are using MCF7 as a drug target for breast cancer testing, so it's perhaps that you will not be able to identify a strong drug target result. So because of the issue with the variability in the MSEP7 dataset from ASCCT that we mentioned previously, we also validate our analysis with another MSEP7 dataset, and this is from a single study obtained from the Gene Expression Omnibus database with 351 samples. So the consensus analysis between the MSEP7 dataset and human breast cancer datasets show a lot stronger signal. And I think one lesson that we learn from this type of analysis is that it is very important to have well-curated data for the analysis. The clustering dendrogram for MSEP7 from the Gene Expression Omnibus dataset also have much clearer signal. In summarize the data we presented so far, we think that MSEP7 cell has to be used extremely cautiously as a model for breast cancer because of its difference in the transcriptomic landscape compared to breast cancer tissue. For example, we show that there is minimal overlap between gene expression in gene expression level between the MSEP7 dataset and breast cancer dataset, and also in terms of druggability of gene target, there's apparently a lot of difference between these two datasets as well. And another thing that we learned is that big data is not necessarily better data. So I think the most important thing in data analysis is to be able to process and obtain data set that work well for the purpose. And there's a lot of things that we would like to do moving forward for this project. For M1, we would like to perform functional annotations for the modules in the M7 dataset and the human breast cancer dataset to make sure that the cluster we saw have biological meaning. And for the second M, we also would like to identify some subnetwork within each network to see what specific difference present within MSEP7 and breast cancer tissue data sets. And in conclusions, I would like to present a quote from my advisor, which is that there is no good science in bad models. And it's really important for scientists to think about what is a good options in terms of designing their experiments. And thank you very much.
Okay, thank you so much for that presentation. A lot of good lessons in your conclusions that I think will contribute to the field. I don't see any questions coming in. Just a reminder to type your question in the question module on the GoToWebinar control panel. I have a question that you may or may not have an answer to, but it just occurred to me as I was thinking about your presentation. Are there, this analysis could be done in other types of models. So you could think about, liver cell line versus the primary liver cell tissue, or gut epithelial cell line versus gut, primary tissue. Do you think that there might be priorities to do these kinds of comparisons in other types of models, and what would those priorities be?
Yeah, I think that's a great question. Thank you so much. So yeah, I think it's important to have priorities in determine which cell model is a good model for clinical research. And I think it depends on the area. So I would think that it's important to analyze the relevance of cell line that is already presented in cancer cell line panels, or also for toxicology testing. It's important to prioritize cell line that is already included in the toxicology screening.
Yeah, that's a really good point. Thank you. We have another question now. How did you obtain the consensus modules?
So for consensus module, we use the algorithm from the weighted gene correlation network analysis, and we just follow the parameters that is in the network. And I will be happy to share the tutorial for those analysis with the audience after the presentation.
Okay. When you compared the two cell models between Brown and Johns Hopkins, did you standardize cell density?
Yeah, I think, so cell density was standardized between those two, and we follow many standardization protocols when we do that analysis. That study was part of the human toxin project, so the goal was to really follow a lot of a quality control method to make sure that the result is rigorous.
Okay, and sort of along these same lines, very nice talk, and may have missed this, but do the MCF7 and breast cancer samples have a similar baseline in regards to p53 status, nuclear receptor densities or sensitivities, antioxidant capacity, etc. Just wondering if one cell type is in a different homeostatic state versus the other.
That's a very interesting question. So MZ7 cell line is used for breast cancer results mostly because of its estrogen receptor positive status. And there are, as mentioned before, many subtypes within breast within MZ7 cell line, and each of these might have different degrees of in terms of the metrics that the audience just mentioned.
Okay, great. And one last, two more questions. Okay, we're going to hold it at 2 so we can get to the next presentation. But in real world stats, what percentage of BRCA1 related breast cancer are actually ER positive? And we know that ER positive breast cancer is more prevalent. Will this affect your conclusions?
So in terms of the percentage, I don't have the number exactly, but as far as I know, like most MZ7 cell line are estrogen positive, estrogen receptor positive. Mm-hmm. And what's the second part of the question?
It's more of an observation. We know that ER positive breast cancer is more prevalent. Does that affect your conclusion?
Yeah, I think that is a good point. So for breast cancer, there is different subtype within like the population of breast cancer patients. So moving forward, one thing I would like to do better for the analysis is to subset the breast cancer data set. So it will only include breast BRCA positive, so that there is more consistencies within that data set.
Okay, great. And then just a last question. Looking ahead, what is your suggestion for future toxicity testing with cell lines? So, you know, what do your conclusions say about how people should be using these models?
So moving forward, I think it's important to use a variety of cell lines for one endpoint to really get the coverage of the in terms of human relevance. And I think there's also a lot of models that are currently available, like organ on a chips, 3D models, and so on. So those models are although relatively new, might present stronger relevance to human physiology.
Mm-hmm. Great, okay. All right, well, thank you so much, V, for your presentation. And we will now move to the second presenter. That is Ignacio Tripodi. Ignacio is a computer science PhD candidate from the University of Colorado Boulder, who joined the department via the interdisciplinary quantitative biology program. His research has involved a combination of genomics, machine learning, and semantic knowledge representation, finally focused on mechanistic inference. He holds a BS in computer science from the University of North Texas and has extensive software engineering industry experience. So we can see your slides, so please go ahead, Ignacio.
Okay, now you guys can hear me. Thank you very much for the introduction, Christy, and thanks a lot for the ASCCT for the opportunity to share my work with you guys. As the title said, I've been working a lot on mechanistic inference, which is a way of saying, you know, I'm interested in automatically inferring the modes of action of compounds of these chemicals that we know are toxic already. So the first line of testing, you know, of course, we via computational or in vitro model, we can predict whether a chemical is toxic or not. But the question I'm the most interested in is why. You know, why is this chemical toxic? What's the mechanism of toxicity? And you guys don't need much convincing about why this is important. but this is very useful not just in the regulatory setting, but also in the small molecule development pipelines, different stages. And what I've been focusing on here was more on the high-level mechanisms of toxicity. So not necessarily on enriching adverse outcome pathways, but rather inferring mechanisms of cellular toxicity. So I just picked this random example from the AOP wiki, which this is pretty well crafted, very detailed AOP that shows, inhibition of certain mitochondrial complex in a certain cell type, leading to Parkinsonian motor deficits. In our case, what we'll be interested in inferring from these experiments is mostly this part, more the cellular effects, and to some extent maybe the molecular initiating event. But in this case, we will be interested in inferring, oh, this is mitochondrial mediated toxicity as a first stage of hypothesis generation. So to this end, I presented a hypothesis generation tool called MexPy. And what MexPy does is mainly two things. It produces predictions of the three most likely mechanisms of toxicity, given a time series of omics assays, and also produces a putative explanation for each of those mechanisms. And this, as you will see, this was performed with transcriptomics assays in this case, but it can really be used with any kind of omics assays, like proteomics or nascent transcription, detecting transcription factor changes over time. And it's also grounded in the open biomedical ontologies. And to this end, I curated 11 different mechanisms of toxicity, mainly from this textbook, where this consisted in essentially turning an entire chapter like caspase-mediated apoptosis to, okay, which are the high-level sequence of events that best summarize and best describe this mechanism of toxicity. And these, of course, are not chosen arbitrarily, but they actually map to, in this particular case, they map to gene ontology, biological process concepts. So we can define a mechanism of toxicity by a series of causally linked gene ontology concepts in this case, or any other ontology that suits better. And this is the entire list of the mechanisms that I curated. Of course, I'm not going to read this aloud to you, but they will be available on the slides later if you want to check out the slides. But just to give you an idea of the variety of mechanisms that I was looking for in this time series. So I thought it would be best to first show by an example of what the output from MexPy is, to show you exactly what I'm trying to accomplish here. In this case, this is a public data set on acetaminophen that was used to treat hepatocytes at a 500 micromolar concentration, and for which we had 4 different time points, for which we got microarray data. So the idea is that after running this through MexPy, comparing the control to the acetaminophen treated cells, we end up with a set of predictions like this. And this is from a real data set. So these are the 3 mechanisms of toxicity that were predicted by MexPy. And one of the reasons we offer more than one is that, as you probably know, for the same chemical, you can have multiple possible correct mechanisms of toxicity, which depend on the cell type or depend on the dosage or other factors. So this is nice because the first 2 are both known mechanisms for acetaminophen, but we don't just provide a score and the list and end up there, as I mentioned. If you take, for example, the second one, which is disruption of calcium homeostasis, we also provide an explanation for it. So this is a narrative that is automatically generated from a knowledge graph and from the experimental data in this time series, which describes a putative explanation for this mechanism. And we see that for this particular mechanism, we see which are the genes that in order are enriched and how they relate to the different mechanism steps. And we also provide a list of the suggested knockouts that you can use to experimentally validate some of these claims, or all of them. And this is particularly useful for more of a pharmacovigilance study, we also provide related variants associated to these key genes that were changing, which may affect the way that an individual will respond to this particular mechanism, by either being more or less susceptible to this mechanism of toxicity. And we also provided a graphical description like this, which this is essentially the same information that you saw on the previous slide in text, but in a graphical form, which you can read from top to bottom. So the nodes on dark gray on the left side are the genes that were experimentally detected to have a significant change in expression, and the purple nodes are the steps of this particular mechanism that have known links. This is all coming from human curated data on human assays. And for some of the nodes, like you see that second node that has a double circle, we also show that these are genes that are known to have a significant level of activity perturbed conditions in this particular tissue type, to provide some more information about this. Of course, that's a nice example because it actually fits on a slide. What I wanted to show here is that there are many of these other explanations that are more comprehensive than the one I just showed you, and they have a lot more information about the different changes in sequence. So even though there's no time to go over all the computational details about how this works, I wanted to at least give you a big picture of how we construct these predictions and these explanations, and just highlight a couple of these components to tell you a bit more about them. We start with a knowledge graph of biomedicine and molecular biology, which is composed of a semantically consistent integration of open biomedical ontologies. So we have the gene ontology, the protein ontology. I will show you some of the inputs in a minute. And also some public databases like the CTD or UniProt. And what we do is we take this graph, we deductively close the graph, and then we generate node embeddings from every node. And this is a vector representation of a concept in this knowledge graph. And I will show you a little bit of how those are composed. Then we take our time series of, in this case, gene expression assays. And by comparing each set of replicates, we come up with the list of genes that are significantly changing at each time point. And the nice thing about this is each of these genes is represented by a vector now from the knowledge graph. And also each of the steps of every mechanism is also represented by a vector. So using a series of calculations involving linear algebra and other types of computation, we generate a score for each of those 11 mechanisms of toxicity. And for the top 3 that were both statistically significant and scored the highest, those are the ones that we choose to generate a narrative and a diagram for. And those are produced using the genes that were changing, only the top genes, and the original version of the knowledge graph. And this is the knowledge graph I used for this was a branch of the Phenolator project, which is led by Tiffany Canahan, one of my collaborators. And it's a project I also collaborated with. And I incorporated a few more edges to this graph that are more relevant to toxicology. So we have an incredible amount of information in this graph, from proteins, genes, chemicals, disease, pathways. We have the cellular toxicogenomics database. We have edges curated from the AOP wiki, and this allows us to have representations like this, where there's a tremendous richness of information that can be conveyed in these knowledge graphs. Like in that, if you look at that dark purple node, we have a pathway, a biochemical pathway that we show how it's related to human disease and which are the genes that are known to participate in that pathway. And also this has part in a more general biological process, which is itself a subclass of an even more general biological process. And by deductively closing, what I mean is that where the relations allow, we can, like in this case, insert new edges for transitive relations, which allows us to have even more relations to generate the embeddings from. Now, moving on to the embeddings, it was clear to me after presenting this a few times that This is something that is not so widely known outside of the computer science audience that specifically works in this. So I thought it would be useful to spend a minute or two just talking about embeddings in general. Essentially, what I'm using here is a numerical representation of each of the nodes in this graph. And this is an idea that originally came from NLP, where these embeddings are created for words rather than concepts. So if you have a word like king, for example, you see there are many sentences that you can train this on that include that word, and based on the adjacency of other words, so based on the neighboring words on either side, and using a neural network, you can come up with a representation, numerical representation of this word. So imagine this is only in 2 dimensions. So we're representing this in a 2-dimensional embedding. We can also there are two really important concepts here. We can perform interesting algebra with these vectors to make inferences, and also we can deduce which concepts are similar to each other based on orientation in this embedding space. So imagine we have, for example, this word king. We subtract the vector for man, we add the vector for woman, and the resulting vector is something that has a very, very similar orientation than the embedding for queen. So what I'm trying to show here is that there's some of the semantic meaning encoded in these numerical representations. Why is this important? Because I use this to generate vector representations of the nodes instead. This is not the same thing, but it's very similar. We just use a number of random walks from every node, and based on those walks, using an algorithm called node to vec, and instead of 2 dimensions like that example, I'm using 32 dimensions to represent each of the concepts. And these are actual nodes from the graph in a Disney plot that I'm showing you how things with similar meaning tend to group together. So for example, you have breast cancer is actually pretty close to neoplasm or BCRA1, and not so close to that cytochrome P450 gene, which is itself close to an oxidation process. So there's meaning encoded in these vectors. For the inputs, I used almost 240 time series from a wide variety of tissue types and a variety of sources, just to make sure that this wasn't overfit to a particular cell type or chemical family. And this included about 150 or so chemicals, out of which we knew there is a somewhat well-established mechanism of toxicity for about 3/4 of them. And using Lima, I performed differential expression across the time points in the time series, and only picked the 100 most significant genes that were changing. And remember, for each of those genes, each of these are denoted by a node in the graph, which means we also have a vector representation of them. And I won't go in much detail on the actual calculations here, but I just wanted to highlight a couple of points from the mechanistic inference step. One of them is that we use cosine similarity to compare all of the gene changes as a whole in a single time point. This is for the more computational savvy. This is essentially the hyperdimensional centroid of all those gene representations. And in this case, where we have 3 time points and we are trying to find enrichment for one mechanism with 5 steps, we calculate the similarity between those centroids and each of the embeddings for the 5 steps of the mechanism. And the second thing that I wanted to point out is that we also, I also came up with this sequential penalty to ensure that the enrichment that we are calculating is also taking into account the sequential order in which these gene changes happen. So we're not just performing a pathway enrichment, we are actually taking into account the order in which these genes are changing to make sure that we reach the correct pathway. And through another series of calculations, we come up with a final score for the mechanism as a whole and perform statistical significance based on random gene draws from the same microarray chip. And since we can have more than one correct answer, it was a little bit difficult to assess the performance, so I had to resort to precision. But I tried to stratify this data in every possible case that I could to show how these, all the different stratifications perform as opposed to a random guess of 11 mechanisms. And one of the problems with these precision values is that these are actually lower bound estimates, because many of the time series that I was using for public data, and this is particularly true for the open TG Gates data, were from low doses of exposure, which means that at that low dose, we may not even be eliciting a toxicity, a toxic response to be able to detect the mechanism properly. So there are a number of false negatives included in these calculations. But to me, the most meaningful one is the middle one, which is the highest dose per chemical and cell type, which means that at that point we have a certain degree of confidence that we are in a toxic response scenario. So for those, about 84-85% of the time, one of the three mechanistic predictions that we propose are coincide with the ones that are expected for that chemical, for the ones that we know the mechanism of toxicity. And finally, I wanted to mention that for those chemicals where we don't know the mechanism of toxicity, this wasn't just a theoretical exercise, and we were able to actually validate a couple of predictions for those. So we took two drugs. One was adapine, the other one was chlorpromazine. And for both of those, MexPy was predicting for the mid and high doses every time, different concentrations. It was predicting the mitochondrial-mediated toxicity as the mechanism of toxicity. So we decided to go ahead and test this. So using the same tissue type and using the same concentrations from those public data sets, we got a mitochondrial damage test kit, and indeed, we showed that at those concentrations, these two chemicals were eliciting a mitochondrial-mediated toxicity, which was great to see this validated on real data. And finally, I wanted to acknowledge the people who helped with this. Tiffany, who was driving the later project for the knowledge graph. Nayland, who's an undergrad that helped me come through many publications where we were trying to classify which are the expected mechanisms of toxicity for these 150 or so distinct chemicals. Jessica, who performed the experimental validation. Robin and Larry, my PIs. And the Tox department, the medical campus, who were very helpful in discussions, curating these mechanisms and providing some cells, as well as my funding and the Biofrontiers Institute. And with that, I'll be happy to take any questions.
Great, thank you, Ignacio. We are open for questions. Please type your question into the question module. I can ask while we're waiting. I and lots of others have been involved in sort of trying to discover and create adverse outcome pathways in order to grow this sort of AOP network that the OECD hosts. And I'm just wondering whether this work could help with sort of hypothesis generation of putative adverse outcome pathways to feed into that system.
Yes, absolutely. So one of my goals, for the next big steps for this would be to have an ontologically grounded representation of these approved, OECD approved, adverse outcome pathways. There is an effort that started with this on the AOP wiki, and it was great. But what we need is an actual representation, the way that I was showing, tying actual events to have this grounded in the ontologies, so to perform, an enrichment of this type. And even there are a lot of new tools, relatively novel tools that came up in the last year or two. One is called GoCam. It's the gene ontology causal activity modules, which allow you to create new concepts as a combination of other concepts. So for the problem with AOPs is that there are events that are very specific, and we don't have an ontology concept for that, like this phosphorylation of this site, of this protein. But you can come up, you can use tools like this to come up with a node that represents this event. So that will allow us. I mean, it's a rather tedious, manual curation process that will require help from the community. But once we have that, the opportunities for finding not just enrichment, but also finding which other genes or proteins that we may have been ignored that are related to a particular step are relevant, it would be a tremendous help. So if you may be interested in helping curate some of these, please contact me, because that will be, that will be it will be helpful as a community effort, and it will be extremely useful.
Great, it's perfect. Perfect call on a webinar. Okay, so a couple questions coming in. Complimentary on your excellent presentation. Just wondering whether the concentration of acetaminophen you're using, 500 micromolar, is not providing overwhelming toxicity. In their research, cell stress and GSH depletion was already occurring far below at sort of 10 to 60 micromolar.
Yeah, it could be possible. I should clarify this. So other than the chlorpromazine and the adapine tests, all the data that I was using was from public data sets. So these are time series that are already being generated and made publicly available. So the downside of that is I had to use whatever was available. Yeah. So yes, some of there are some exposure doses that are actually even more ridiculous than that. There was an acetaminophen exposure of 1,000 micromolar. And the other problem with any kind of time series analysis like this is also the choice of time points. I suspect that for many of the receptor type mediated toxicity, the time points that are generally chosen, which are standard like 12, 24 hours, 48 hours, may be way too late. And we actually need sooner time points shorter time points to detect those changes, or maybe even a different assay altogether. But yes, I agree that some of this concentration, and that is also reflected in some of the precision. I wanted to leave this as true to the data, as I could. So even though I think that the precision should be way better than 84, 85%, this is the product of using all these public data sets that they could be either overwhelmingly high in a dose or way too low in a dose.
Yeah. Okay. Just last question, wondering if your work is published, because they're interested in learning more.
Sure. So it's currently under review, but also if you when you get the slides, I also included the title of the paper that is under review, and there's also a preprint in bioRxiv. So I don't know. I can guarantee it's going to be the same, content that will be published, of course. But you can if you look for that title in bioRxiv, you will be able to find a preprint and see a better description of all the calculations involved and all the listing of all the data sets that were used in all these different issues.
Right. Well, I will connect you with that questionnaire, so then you can engage afterward.
Perfect. Yeah.
Well, I think we're going to go ahead and end the webinar. Oh no, we have one last question. What is the valorization potential of your research?
What do you mean valorization?
I'm
potential?
Not sure.
Exactly. Like commercial valorization?
Maybe that could be it. Let me see if I can. Let me see if I can unmute the questioner. Eva, you are unmuted. If you want to go, maybe explain a little bit your question.
Okay, can you hear me?
Yeah.
Yes.
Okay. So I was wondering, because MaxPy, that is a thing that you're using, right? MaxPy. It seems like that you can potentially use it like for a lot of toxicity testing, as you really extensively explained and really good explained. But maybe you can even bring it further to like other sort of industries, and then I don't know, put a patent on it or some kind. What is your opinion about this?
I would prefer. I mean, that's a tough question. Probably beyond the scope of this webinar, but at this moment, I think I would prefer for this to be publicly available, so that it can be used by the largest amount of teams, not just in the industry, but also in the academic setting, and especially in regulatory environments. I'm sure there are a lot of, as you can imagine, there are a lot of possible extensions to this. Maybe there's an option for a even more comprehensive version of this that can be distributed as a commercial option. But so far, this is a GitHub project, which is public. All the code is open source, and yes, I'm not at the moment in the process of getting a patent on this.
Okay.
Thanks for the discussion, for the question, and just want to thank all of you for listening. And I would especially like to thank both Ignacio and V for their presentations, and congratulate them for winning the awards that they won last fall. Just a reminder that anyone who participates in each ASCCT meeting every year is potentially eligible. And so I'd encourage you all, once the call for abstracts comes out, to participate. Yeah, thank you very much, and have a great day, everyone.
Thank you.
Bye.